Welcome to one and all. The topic I have chosen for discussion is enzymes. Enzymes are the complex catalysts produced by living cells with a high degree of efficiency which permits biological reactions to occur rapidly through a well-defined pathway. They accelerate the rate of reactions without being lost in the process. Enzymes are located in the cell's cytoplasm, mitochondria, tissues, and body fluids. Properties. Enzymes acts as a catalyst. Enzymes are proteins. Specificity. Enzymes can catalyze only a small range of reactions or only one reaction. Types of specificity. Absolute specificity. Enzymes under this group catalyzes only one particular reaction. Group specificity. Enzymes under this category acts on a group of substances possessing a particular type of linkage. Optical specificity. Enzymes under this catalyzes same group of substances possessing same optical activity. Purification methods. Purification is done in order to isolate the enzymes for analytical and research purposes. Methods involved. Particles of smaller molecular weight and electrolytes can be removed. Precipitation. Precipitation with alcohol, acetone and neutral sulfates can also be removed. Adsorption. Adsorption by adsorbents like kaolin, animal charcoal and alumina followed by elution method. Separation and isolation of enzymes by chromatography is a well-known method. Classification of enzymes. Enzymes are classified into six major groups. They are oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Oxidoreductases. They catalyze the oxidoreduction reactions between two substances. It involves transfer of hydrogen atoms or electrons from one substrate to another. Example for oxidoreductases are oxidase and peroxidase. Transferases. Transfer of certain groups from one organic compound to another. Example, transaminases. It involves transfer of amino groups to keto acids. Transacylases. It involves in transfer of acyl and acetyl groups. Transpeptidases. It involves in transfer of amino acids or peptides from one peptide to another. Hydrolases. It catalyzes the hydrolysis of substrates having large molecular weight. Example, carbohydrases which splits carbohydrates, esterases that splits ester linkages. Lyases, which catalyzes the addition or removal of some chemical groups by mechanisms other than hydrolysis, oxidation, and reduction. Isomerases. Enzymes that catalyze the interconversion of isomers comes under this group. Example, epimerase. It catalyzes the L-alanine to D-alanine. Ligases. It catalyzes the linking together of two compounds coupled with activation of ATP or GTP. Example, acyl-CoA ligase and succinyl-CoA ligase. Mechanism of action. The compound on which the enzyme acts is called a substrate. Enzymes, being proteins, have reactive sites on their surface on which the substrate fits. All enzymatic reactions involve temporary union of substrate with enzymes. The substrate just fits on the reactive site to form an enzyme-substrate complex, also referred as Michaelis complex. Then activation of substrate takes place. The combination is called as the activated complex. This combination weakens the structure of substrate and the bond in the substrate is strained to rupture. When the bond ruptures, the reaction products are liberated into the solution and the original enzyme molecule is regenerated. This enzyme molecule can now catalyze fresh substrate molecule and the process will be repeated. Coenzymes. Most enzymes require the presence of certain non-protein compounds for their action which help in accelerating the enzyme action. In contrast to enzymes, they are heat stable and of smaller molecular weight. They are called coenzymes or prosthetic groups. If the activator is firmly attached to the enzyme protein, it is called a prosthetic group. If the non-protein compounds are not firmly attached to the enzyme protein and exist in free state in a solution, they are called coenzymes. The entire enzyme system consisting of enzyme, protein and the coenzyme or prosthetic group is called holoenzyme. The protein portion is sometimes called an apoenzyme. Examples for coenzymes are thymine pyrophosphate, nicotinamide adenine dinucleotide, nicotinamide adenine dinucleotide phosphate, flavin mononucleotide, and flavin adenine dinucleotide. Thank you. Have a nice day.